Welcome to all of you who are joining our webinar today. So I'm the president of the French Society for Cell and Gene Therapy, and the board and myself are very happy to present to you in 2023 a series of webinars on gene editing and gene therapy, a technology that we believe that in the future might lead to very powerful therapies. But we are not there yet. So we insist as a society and our board to give the opportunity to young researchers, so like PhD students or postdoctoral students, to present their work to the scientific community and, of course, beyond to anybody that is interested. Therefore, it is my pleasure for the first webinar to introduce to you Juliette Rosier. She just has finished her PhD in the Bordeaux Institute of Oncology at the Inserm 1312 and in the group that is called BioGo under the supervision of François Gaudry. So she worked during her PhD actually on the optimization and the increasing of the safety of gene editing tools for disease treatments. So she's definitely the right person to give you an introduction to the gene editing tools for gene therapy applications. So as you can see, you can ask questions via the chat, which I, at the end of the talk, will then ask to Juliette. So I hope you enjoy it, and please go ahead, Juliette.
Well, hello everyone. I thank you, Els, for this nice introduction. As she just told you, my name is Juliette, and I've been working on CRISPR-Cas9 for my PhD at the Bordeaux Institute of Oncology. I'm going to talk to you today about genome editing for gene therapy applications. And first, I'm going to briefly remind you the basic principle of genome editing. Humans are constitutive of billions of cells that contain in their nucleus the DNA. And sometimes the DNA contains a mutation in its sequence, leading to defective genes that can be responsible for a genetic disease. So the idea of genome editing is to target the defective gene in order to disrupt the sequence and remove the mutation in order to resynthesize correct DNA sequence and therefore to cure the gene and cure the disease. So in order to do that, we have to use a tool that's able to enter the cells and recognize the DNA to cut it. So the first tools able to cut the DNA were identified in the 1970s, and they were restriction enzymes. These are nucleases that recognize a specific DNA pattern to induce the cut in the DNA. However, they are not highly specific and they are not used in genome editing. They are widely used in molecular cloning. The next tool developed for gene targeting was zinc finger nuclease in the 1980s. They work as a two-component system formed by a nuclease and a zinc finger DNA binding domains. They work as a dimer and they recognize a 3-base pair site recognition on the DNA. They are still not highly specific and the efficiency is compromised. So the next tool developed for genome editing were TALENs for transcription activator-like effector nuclease in 2010. They work in a similar way as the ZFN technology, as they also work as a dimer using a nuclease, but the DNA binding domain is different and they use TAL effectors. The amino acid repeat possesses single nucleotide recognition site. This technology increased the targeting capacity and specificity. However, the time and the cost for the development was really complex. And even if they showed a great improvement for genome editing, the complexity for their design really prevented the widespread applications. So it wasn't really until the development of the CRISPR-Cas9 technology that really induced a great revolution in the molecular biology field, as this system is really cheap compared to the others, really easy to use, and in many different applications. So a brief history of this CRISPR-Cas9 system. In 1987, it was the first time that unusual sequences were discovered in bacteria. And these sequences were called CRISPR in 2002. And the implication in adaptive immunity of bacteria was established in 2005. In 2012, the team led by Emmanuel Charpentier and Jennifer Doudna used this system, these CRISPR sequences associated with a protein called Cas, to specifically modify the genome. A year later, this system was firstly used to modify eukaryote cells. That's really amazing, is that only 3 years after the first use on eukaryote cells, it was the first clinical trial for human lung cancer using CRISPR-Cas9. This system was of great interest for the scientific community, and researchers worked on optimizing the system to produce more specific and more precise tool in order to develop technologies such as base editing and prime editing. In 2020, Emmanuel Charpentier and Jennifer Doudna received the Nobel Prize for the discovery of CRISPR-Cas9. So how does it really work? Well, CRISPR-Cas9 is a two-component system. There's a CRISPR part and a Cas9 part. The CRISPR part is an RNA that we can design to recognize a specific sequence in the DNA. So it works as a guide. And the Cas9 part is a protein that works as scissors because it has two catalytic domains called HNH and OFC, both able to cut one strand of the DNA. So these two components associate into a ribonucleoprotein complex, the CRISPR-Cas9. Therefore, the guide can lead the Cas9 to the specific DNA target next to a 3-nucleotide long sequence called proto-adjacent motif PAM. And when the CRISPR-Cas9 has recognized the target, the Cas9 is activated and undergoes a conformational change, and both catalytic sites become active, and therefore that leads to a double-strand break of the DNA, or DSB. This double-strand break is therefore repaired by cellular mechanisms, either by non-conservative mechanisms called non-homologous enjoining. That's when nucleotides are reorganized to reform the DNA sequence, but randomly, and that generates insertions or deletions referred as indels. And that can lead to a frame shift of the sequence and therefore gene inactivation. And it can also be repaired if we provide a template that contains the sequence we want to introduce in the DNA. The cell can repair through homology-directed repair mechanisms, HDR, and that leads to a precise gene modification and therefore gene correction. So in order to perform the genome editing, we have to bring the CRISPR-Cas9 system into the cell, into the nucleus to reach the DNA. So we have to use some vectorization systems to bring the CRISPR-Cas9 to the target. Among this system, there have been the viral vectors that have been used, such as lenti-vectors and adeno-associated viruses. Lenti-vectors are highly efficient with a high packing capacity. However, they can integrate in the genome, so there's a risk of insertional mutagenesis, and also they lead to permanent expression of the Cas9, which is not preferred. And the AAV vectors are also highly efficient. They can be pseudotyped to broaden the cell tropism, and they lead to a transient expression of the Cas9, which is preferred. However, their limited size and complex production prevent their use in genome editing. So beside viral vectors, as they are really highly effective, their risk is limits their applications. So they've been non-viral methods that's been developed. And for non-viral delivery, we can use different biomolecular formats of the Cas9. The Cas9 can be introduced as DNA, as a plasmid, or as RNA, or directly as an RNP complex, where the Cas9 is in protein form. So for all these three different biomolecular formats, there have been many different non-viral methods developed. I'm just gonna present you a few. For instance, there's the physical methods, such as electroporation. That's when we apply an electric shock to the cells to disrupt the cell membrane and therefore allow the CRISPR-Cas9 system to enter the cell. There's also microinjection. That's when we directly inject the CRISPR-Cas9 system into the cell through a needle. It's highly effective. However, we can only edit one cell at a time, so it's used for embryo research. And besides physical methods, we can also use chemical methods, such as lipid nanoparticles, also widely used. That's when the CRISPR-Cas9 system is protected by the liposome to enter the cell through endocytosis. And we can also use cold nanoparticles that can be functionalized, but also polymer nanoparticles or cell-penetrated peptides. And a new tool also developed in 2021 still enlarged the toolbox available to vectorize the CRISPR-Cas9 system. These are called the nanoblades. They are virus-like particles, so they lack the viral genome, so they cannot replicate and they are not infectious. But we have fused the GAG protein to the Cas9. So and there's also a proteolytic site, so that once the nanoblade penetrates the cells, a protease can cut the proteolytic site and therefore release the CRISPR-Cas9 RNP into the cell. These nanoblades can also be pseudotyped with envelope glycoproteins so that they can target many different types of cells, and most importantly, they can target clinically relevant cells. So the high efficiency and the low cost of the technology, and also the great diversity of tools available to vectorize the system makes it really easy to use in many different applications. And it's used, for instance, in agriculture for plants modification, for drug screening, also for embryo research, to generate animal models. But I'm just gonna talk to you today about a few examples for the use for the treatment of human diseases. So it presented as an alternative to gene therapy with no insertional mutagenesis and a physiological gene regulation. And it's already been used in many clinical trials for cancer treatment and for the treatment of some genetic diseases. For cancer treatment, it's used for the generation of CAR T-cell therapy. The T-cells are transduced with an ROV expressing a core antigen, and with CRISPR-Cas9 targeting the T-cell receptor and PD-1. So that allows to generate a CAR T-cell and to avoid graft-versus-host disease, and also that allows to reverse the immunosuppression of the cancer cell towards the immune system. That's used for malignant hemopathies and solid tumors. However, for solid tumors, their minor accessibility makes it still difficult to use this kind of treatment. For the treatment of some genetic diseases, there's an example for ex vivo treatment of beta hemoglobinopathy, such as sickle cell disease. In sickle cell disease, the locus of interest is the beta globin gene cluster located on the 11th chromosome. It's composed of 5 genes that are successively expressed during the development. The HBG genes are responsible for the production of gamma globins involved in fetal hemoglobin, and the HBB gene is responsible for beta globin chains involved in adult hemoglobin. This adult hemoglobin is non-functional in sickle cell disease. So there's a repression on the HBG genes due to transcription repressor coded by BCL11A on the 2nd chromosome. So the clinical trial strategy is to target this BCL11A gene using CRISPR-Cas9 in order to suppress the repression it plays on the HPG genes. Therefore, that leads to an increase production of fetal hemoglobin, which can restore the phenotype of the patient. Another application of the CRISPR-Cas9 technology, but this time for in vivo treatment, is, for instance, the Leber congenital amaurosis type 10. In this disease, the CEP290 gene is mutated in the 26th intron, leading to an abnormal splicing and therefore premature stop codon. So the clinical trial strategy called EDIT-101 uses an AAV to enter the CRISPR-Cas9 system through a subretinal injection to target both ends of the mutation, therefore to remove the mutation to restore a normal splicing, and therefore that restores the production of the protein. Another successful clinical trial for in vivo treatment of CRISPR-Cas9 is the transthyretin amyloidosis. In this disease, there's an accumulation of a TTR protein in different tissues. And this protein is produced by the liver. So the clinical trial strategy uses lipid nanoparticles to bring the CRISPR-Cas9 system through an IV, because IV drugs are treated by the liver. So that allows a rapid liver distribution and a knockout of the gene in the liver, and that reduces the concentration of the TTR protein. So even if CRISPR-Cas9 has shown great promise for research and for clinical trial, we still have to keep in mind that it induces some definitive genome modifications. So it can sometimes not always be accurate, and that can lead to a genotoxicity with the generation of unwanted genomic byproducts. Among this genotoxicity, there's the off-target genotoxicity. That's when the Cas9 cuts at a different site than it was first intended because of either a mismatch or homologous sequence. But these effects are really well described, and there are many tools that exist to predict and detect them. And also it's resolved by variants of the Cas9 that have been developed to make it really much more specific and high fidelity. Besides this genotoxicity, off-target genotoxicity, there's also the on-target genotoxicity. That's when the Cas9 does cut at the desired locus. However, the double-strand break isn't repaired as expected, and that can lead to undesired or unexpected genomic byproducts. This genotoxicity was still underestimated until a few years ago, and it still has to be evaluated to be understood and then prevented. What we have to remember is that the genotoxicity is due to the double-strand break in the DNA. So strategies have been developed to modify the CRISPR-Cas9 system in order to make it independent of the double-strand break. For instance, the CRISPR-Cas9 nickase have been developed. That's when one or the other catalytic site is inactivated. Therefore, only one active site remains, and it leads to only a single strand break, which isn't repaired by the same cellular mechanisms and should not lead to any genotoxicity. Also, another improvement of the CRISPR-Cas9 system has been the base editing technology. That's when a nickase is fused to a deaminase, either adenine deaminase or cytosine deaminase, so that it can convert an A to a G or a C to a T. But at first, this was not possible to do transversions and only transitions. And now, even though it's been reported some transversion can occur, it's still difficult. And this technology is really effective, but only for purposes of gene conversions. So a new tool was also developed to provide a more wide range of gene modifications possibility. So it's called the prime editing technology. A nickase in this case is fused to a reverse transcriptase, and this time the guide used is used is actually a PEG RNA, which can at the same time guide the system, and it contains also a primer binding sequence, the RT template, and the edit sequence we want to introduce. So it creates an all-in-one system that can target the sequence and introduce the desired modification at the target site. However, the design is really complex, and for now, it's not yet effective in stem cells. So to sum up and to conclude on the CRISPR-Cas9 technology for genome editing, it's a really highly promising technology with a great future in gene therapy. It's a really powerful technology, so it has to be managed with care and consciousness. It still has the potential to be improved and optimized, and researchers are working on it in order to provide a really safe and more efficient gene therapy tool that can be used for research and that can be used for clinical trials in order to treat the most genetic diseases and cancer. So I thank you for listening. I'll try to answer your question, and I'll let Els present you the next webinar, or I'll cut my...
Thank you very much, Juliette. I'm really happy with this beautiful presentation and introduction to the gene editing webinars. Indeed, in the next webinar, we will have Laura Ugalde Diaz from Madrid, who will talk about gene editing and how it can be applied for a specific disease like Fanconi anemia. So we can go over to the questions now. So I think you can stop sharing, Juliette, your screen. And you can put on also your, mic. So I think that this was really a nice overview of how fast some technologies can develop from the first time they were discovered until the transfer into the clinic. I think both, since we are a little bit working on these tools, we know that we can still improve really a lot of things. So one of my questions is, you, for example, showed that the prime editing would definitely be an improvement, but that it's still not very efficacious in the target cells where we need to use it, like hematopoietic stem cells or like immune cells or whatever. Do you think that we could package this system in these VLPs or in nanoparticles? Can you extend a little bit on what people try to do, maybe, to improve this?
Some optimization possible is to fuse the Cas9 fused to the reverse transcriptate into the nanoblades, for instance, and also instead of using a simple guide RNA, we can synthesize the PEG RNA through the nanoblades. So it could really be possible to construct the prime editing technology into the nanoblades in order to target many different types of cells with a high efficiency and no toxicity. That should really be a way of the improvement in the next year.
And I'm not sure that everybody really understood the term genotoxicity or insertional mutagenesis. So I think in the gene therapy field, we all know that, for example, the SCID trials, the bubble children that were corrected with a lentiviral vector. Can you explain a little bit the difference we have between using, for example, a gene addition approach with lentiviral vectors and a CRISPR or prime editing system?
Well, for gene addition, we add the defective gene. We add the correct sequence of the defective genes that can insert in a random way almost random way. And in some case, it can insert next to an oncogene, and that could lead to reactivation of this gene and therefore leading to adverse events. So on the contrary,
it's like development of a leukemia,
yeah,
like they have shown for the SCID children.
Yeah. So the principle of CRISPR-Cas9 is to target the endogenous gene. So we do not add a gene at a random site. We correct the gene at the endogenous site. So that should not lead to insertion or mutagenesis.
Yeah. Plus, the advantage is most probably that we have a physiological expression of that gene compared to when we introduce a promoter with a correcting gene, and this promoter normally doesndoesn't have all the sequences to regulate the expression like when we do CRISPR-Cas9 gene editing, right? So one of the other things I wanted to underline is these are really, I think, for the future, important tools. It's used in IPS strategies. It's used for making CAR T cells more efficient, better. But I think we also, maybe you can just briefly explain how important this technique was for the research area, because this is a tool almost used in every single scientific lab now. Can you just explain a little bit why it is so useful or what we can do with it?
Well, in research, it's really helpful. It's really easy to use, so any students can use it during an internship, and we can modify with a high efficiency any genes we want. So we can study the effect of an inactivation of the gene, of certain modification. We can test many different guides also to select the most effective one. That allows also to generate some animal models with the desired modification with only one generation when we modify the mouse. It's used for drug screening, so we can identify potential resistant gene when we can screen all the genes with a bank of different guides that target the whole genome. We can therefore identify potential genes of interest to cancer resistance or such things.
Yeah, we can actually really see certain pathways in cancer cells coming up that we can then target with certain drugs. I think these tools are an amazing advancement in our field, and although they are already in the clinic, they are constantly improved, and I think we will in the future have really important therapeutic methods derived or with the help of these tools to attack cancer or to correct genetic diseases and so on. Juliette, I thank you really a lot for these clear explanations. And I hope that everybody enjoyed the webinar. And we will see you, I think, in March. The next one is on the 10th of March for the next webinar. Thank you again, and have a very nice afternoon. Bye-bye.